Hello and welcome to El Zoominario. Today we're very excited to have Rosana Zenil-Ferguson. She joined the Department of Biology at the University of Hawaii, Manoa as an assistant professor in January 2019. She earned her PhD in biology from the University of Florida after completing a master's in probability and statistics at CIMAT, Mexico. She grew up in Toluca, Mexico, which is a city above 8,500 feet above sea level, and is located in the Trans-Mexican Volcanic Belt, surrounded by pine oak forests. And the city is very known for being the birthplace of Cerveza Victoria. Thank you very much, Rosana. Please, we're very happy to have you and take it from here.
Thank you, Claudia. It's great to be in El Zoominario. And I wanted to start my first slide acknowledging my two collaborators in this project, amazing scientists, Santiago Ramirez Barahona, who's also Mexican. He works in the Instituto de Biología UNAM, Mexico, and Emily Cesar, who is an associate professor at the Department of Biology at University of Florida, and she's the emerging leader of botany this year. So I don't know if her talk from botany is available online, but if it doesn't, you should talk to her about ferns in Africa. It was amazing. So today, of course, this doesn't advance. I'm going to talk about a lot polyploidy, and I wanted to make sure for those of you who don't work in botany or polyploidy know what it is. It's a mutation that randomly increases the chromosome numbers of a species, and it happens during meiosis. For example, here we have a diploid species with two chromosomes, A, and I put two colors. And during regular meiosis, this cell will undergo two divisions and end up with a gamete that has just one chromosome. But in this is what we call the reduced gamete. But sometimes during meiosis, things can go wrong and a separation cannot occur, in which case we can produce two unreduced gametes if the mistake happens during the first round of meiosis. And it could also happen that the mistake in the separation happens during the second round, in which case, a plant can produce, for example, pollen that is reduced and unreduced, like this example. But the important thing of polyploidy is that using these unreduced and reduced gametes, we can produce new species, right? For example, here, if we had a plant with 10 chromosomes and another plant with 16 chromosomes, and these two produce unreduced gametes, we can ultimately have a chromosome number of 26, and that is corresponding to a new tetraploid and a new species. And this is the example of some of very famous genus, Arabidopsis, in which you mix two species, Thaliana and Arenosa, and you obtain tetraploid Suecica with 26 chromosomes. Not only is interesting in the terms of speciation or including extinction, it's also important in ecology. One of the examples that I like to use because I am in Hawaii is the silver swords. There's a hypothesized polyploidy at the root of the clade right before the dispersal to the Hawaiian Islands. So a lot of people have hypothesized how polyploidy has increased the opportunity for the silver swords to establish and disperse to new habitats in the islands. And there's many examples like this, including Bidens in the islands. But one recent and important study in angiosperms was finding that the frequency of polyploidy increases with latitude. So you can see this map here. The red means more incidence of polyploidy, and you can see that the redder it is, the northern latitude this is. This is from a study from Anna Rice in the group of Itai Mayros from 2019, right? So they conclude that polyploidy frequency is correlated with temperature and mostly was mediated by the composition found in these biomes, whether they are perennial herbaceous or not. So some of the hypotheses that this paper and others have laid out for this pattern are the following. So the first one is the recurrent and reduced gamete production in harsh environments. So a lot of studies have induced polyploidy in sort of extreme conditions, and they have found that the reduced gamete production increases when the habitat or the environment is harsher. The secondary contact hypothesis is that allopolyploids, like the example I show you from Arabidopsis, should be more abundant in formerly glaciated regions where new contacts of divergent populations occur, and perhaps that's mostly towards the poles. The third hypothesis is whether life history traits have some interaction with polyploidy that enhance or decrease the probability of polyploidy events to occur. And I have some work, most of my lab and the current work we're doing is sort of merging these ideas of different traits interacting with polyploidy. The fourth hypothesis to this is that the tropics are phosphorus and nitrogen poor, and they have soils that are nutrient poor. So polyploids that hypothetically have more genome content cannot be sustained. And those are articles that are starting to coming up more and more frequently. And finally, the fifth hypothesis to this is that polyploids might be more frequent in disturbed and vacant habitats because there's more availability and less competition, so they can inhabit these new opening spaces. And I think this distribution is the result of a combination of these five different hypotheses. There's no one that is more prevalent, and I think that's the reason why there's so much work ongoing across all these different hypotheses. But this specific talk is going to focus mostly on this idea of whether polyploids can be more frequent because there's more opportunity in niches with lower competition. And incidentally, the results are going to tap slightly, or at least question, number one hypothesis and number four. So the goal of the talk of this study that I'm presenting is to integrate polyploidy in some ecology, right? Some very basic ecology. And I don't like to say full ecology because I'm not an ecologist. So my perspective is very basic, right? So I'm gonna use as a proxy of ecology biome. And I'm gonna ask whether polyploids are different, or polyploidy occurs differently across different habitats, and how do polyploids disperse to these new habitats. And all this is going to happen under the background of diversification. And the reason why it's happening on the background of diversification is because if we don't consider speciation and extinction, we most likely overestimate or underestimate some of these dispersal parameters or polyploidy parameters. When we're not considering that, for example, diploids could speciate really quickly. And the perfect system to do this is Inference. And the reason why is because they have high incidence of at least recent polyploidy, 37% estimated. And the spores can disperse anywhere. So we don't have some of these local spatial restrictions like sunflowering plants will do, because they disperse thanks to for example, pollination. They are super pollination dependent, or they are their seeds depend on the dispersal of some mammals. So we don't have those situations with ferns. So it makes the estimation of dispersal slightly easier. And for ferns, then we collected a really large data set. So we have a recent time-calibrated phylogeny by Weston Testo and Michael Sondu from 2016. I curated a big data set of chromosome number records using some previous software that I designed with the help of Emily and one undergrad in her lab. And then using a pipeline from Santiago, we did a biome determination for all these taxa, and we found three broad biomes, or we classify three broad categories, just like his paper, his recent paper, that are temperate, tropical, and arid. And so you can see here that we have huge mix of diploid, polyploid species and a huge mix of biome all across the phylogeny. And then the next step to this project was creating a model to talk about the polyploidy itself. And the best model to do this from macroevolution perspective is one that we call the BC model, the binary state speciation and extinction model, that for polyploidy looks like this, right? So it has two states, diploid and polyploid here in yellow. And then it has two parameters for each state, one for speciation and one for extinction. And then a parameter for polyploidy, that is what takes this state diploid into polyploid. And of course, is its extinction and speciation for polyploids. I should mention that there are more complex models than this, which we have fed for polyploidy alone. I won't talk about them right now because I wanted to create a simpler story, but if anyone is interested in more advanced hidden state models, I'll be happy to talk to you later about them. So when we fit this model in polyploidy in the data, what we find is that what other study had found in 2011, that diploids have faster net diversification, and net diversification is defined as speciation minus extinction, than polyploids. And the question here is, who are these polyploid ferns and what habitat do they prefer? Why are they going slower in this process of speciation minus extinction? So then I decided to use this same data set for biome. So this same phylogeny, the same species, and now biome, and fit a similar model, what we call the museum model, but only for the three states of biome. And what you find is that what you will find here is three states: tropical, temperate, tropical, and arid biomes. And then the potential dispersals back and forth from these three biomes and their speciations and extinctions. And what we found very significant is that arid ferns that are in arid biomes do definitely have a slower net diversification than anything else, either temperate or tropical. And the temperate and tropical pretty much overlap in terms of the diversification. So how do we go about the integration of this knowledge of arid environments or in polyploidy, right? And this knowledge of biomes. So the way to do that is by creating a little more complex model that recreates some of these traits, but considering both traits at the same time. And that model is this model. So it looks pretty ugly, but in the top, you can see that all these states are diploid, and in the bottom, all these states are polyploid. And then we have tropical, temperate, and arid environments for diploid taxa. And then for polyploid taxa, we have again tropical, temperate, and arid environments. All these rates here are the polyploidies, and they can be different. They can be different depending on the biome the plant is. And then each one of those has their own speciation and extinction and their own dispersal, but while maintaining their ploidy. So the reason why to build this complex model is because it will allow us to answer some of these questions of the interaction of polyploidy and where the plant lives. And for example, one of those is whether or not polyploidy rates differ across different biomes, across different habitats. So this will be the orange, the blue, and the yellow rate, right? And when I estimate those rates in this model, turns out that polyploidy rate is much, much faster in tropical than in temperate, and both of them are much faster than in arid environments. So this result is a little bit different in what it has been found in angiosperms. In angiosperms, for a long time, it has been hypothesized that temperate environments have faster rates of polyploidy, but no model have actually combined their biome and their polyploidy. So whether it's the same in angiosperms that is happening in ferns, we don't know for sure. But it's really interesting that polyploidy in tropical environments is coming as the fastest rate of all polyploidies. And another thing we can answer with this model is, for example, how do diploids and polyploids disperse to new habitats? And in particular, what's the difference between diploids dispersing from from temperate to tropical, or polyploids from temperate to tropical environments? And when we fit this model and estimate those rates, what we find is that it's much faster to move from temperate to tropical biomes for diploids than it is for polyploids. So polyploids are having a harder time to go back to the tropics. So this might explain some of these patterns of why we observe more polyploidy out of the tropics. So just to finalize my talk, I think it was 20 minutes. This hypothesis of polyploidy being more frequent in disturbing vacant environments due to more high niche availability really it really is important for many things. But one of those is, are we studying this in the context of diversification, and are we allowing polyploidy rates to happen, and dispersals to happen in our models? So the first thing that we found then is that diploids have faster net diversification than polyploids across ferns. And what is missing here still is to figure out if diploids in tropical environments have faster net diversification than polyploids in tropical environments. And I don't present those results because those rates haven't been estimated correctly yet. So we're on the process of doing that. But in both temperate and tropical biomes, we have similar net diversifications for ferns, and they are much faster than arid biomes, implying that arid biomes and diversification, speciation, and extinction in arid biomes is going to be hard, no matter if you're a diploid or a polyploid. Finally, polyploids in temperate environments, the presence of polyploids in temperate environments is not due to more polyploidy. As we saw, there's a lot more polyploidy in the tropics once you've considered diversification. And the question here, in what I said, there's a little bit of implication about the research of unreduced gametes, is like, if there is more unreduced gamete formation in harsher environments, what does it say about survival? There might be more unreduced gamete formation, but that doesn't necessarily lead to more polyploidy because those polyploids are not surviving. So is it true that there's more unreduced gamete formation for ferns? We don't know, because most of those studies have focused again in angiosperms. Finally, polyploids disperse with a slower rate from temperate to tropical compared to diploid. And this may explain a little bit more of the pattern on why polyploids, once created in temperate environments, even though it's a slower pace, they don't move enough. And what's missing here is knowing still the rate of how polyploids from the tropics, whether they move faster back to temperate environments. So we are working still on some of these estimations, but we think this model is going to allow us to answer more of these questions. In terms of future directions, one of the things that I already mentioned is whether that we're missing the estimation of net diversification of ploidy by biome. So whether diploids are having less net diversification in tropical environments than polyploids is something that we still need to test. Not all, the second thing that is missing is that not all the changes in polyploidy are connected to the diversification itself. Some are anagenetic, some can happen without any speciation and extinction. And this is important also for the estimation of dispersal, because these rates are interconnected. Also, just because of our own biases on how we study polyploidy and which habitats we have available, there are a lot of unaccounted polyploids in tropical environments. And I wanted to show you this plot that Santiago made. So here in the x-axis is very small, but you have temperate and then tropical and arid environment. And these are all our samples size for temperate, tropical, and arid. And you can clearly see that the tropical environment, we know a lot less about who is diploid and who is polyploid. We know that plants live there, but we don't know their chromosome numbers. All this purple thing is pretty much unknown, right? So we know a lot more about temperate and a lot more about arid, and this is because we have those biomes closer to North America and Europe, which there's a bias in the study. So there's a lot of bias in the study of chromosome numbers in the tropics. So with that, I want to finish my my science talk, and I'm going to move to what Claudia asked me for this talk. Talk a little bit about where I'm from. So I grew up and was raised in Toluca, Mexico. I come from a family that everybody grew up there and was born there. There was no no one migrating and dispersing, especially not to Hawaii. But this is a very interesting place in central Mexico. It's just 60 kilometers west from Mexico City. So about an hour driving, you arrive to Mexico City. But the important part of this place, Toluca, is that it's located in the trans volcanic Mexican belt. So it has a really complex geological history. And we're surrounded by valleys and volcanoes and hills of all sorts and kinds. And it is interesting because when I moved to Hawaii, I realized that volcanoes inspire people, right? And all my family, all of them, are artists. And you can see some of their artistic representations. We have architects and stained glass designers and jewelers and painters. And it was very interesting to grow in a family that nobody considers science and everybody was doing art, right? But I reached the point where I realized that the reason, why everybody does art is because of the volcano. Just our environment, our surroundings are so important. And this is what we can see from the city. El Nevado de Toluca is the most important volcano. Its name in Nahuatl is Xinantecatl. And it has two lakes at the top. This is elevation about 3,000 meters above sea level. And I always feel like it was weird that I was not an artist and I didn't have any artistic inclination, given that all my family does. But I realized that I somehow do, because what I was seeing as art was this nature, right? So growing up here and going up in hikes, I was able to pick up all this great diversity of flowering and non-flowering and some fungal species. And right now we know that there's at least 30 endemic species in the volcano, all of them in danger of extinction due to agricultural practices, and mostly corn and potato fields, and a lot of greenhouse and pollution effects. So in this talk, just to finalize, I want to mention that there's incredible people in Toluca really working for the preservation of these species. And one of them is this incredible woman, Ana Margarita Romo Ortega, who is now the director of the environment department in the city. And she's been doing incredible programs to rescue some of the fauna and flora of the volcano. You can see here that we have our best representative as a volcano rabbit. But she has been doing also a lot of field work and asking people to do observations in a website called Naturalista MX to rescue some of the species. So if anyone is interested in doing some work or some research in the transvolcanic belt, The Nevado de Toluca is one of the most overlooked areas in central Mexico. So, and Ana Margarita is one of the most valuable people to talk about conservation of some of these species. And with that, I want to acknowledge my collaborators, Emily and Santiago, and the artists that willingly share their pictures for this presentation, and my funding source, that is the University of Hawaii, OVCI. Thank you.
Thank you very much, Rosana. I'm going to stop the recording now.